So first of all, thank you to the organizer for inviting me, and thank you to all of you for listening to me. I'm going to present the work that we have been done in Spain, in the north of Spain, where is Santander, and the work of my group working in listeriosis so far for almost 23 years. And we have right now a possibility of a vaccine for listeriosis, a dendritic cell-based vaccine with a novel antigen that is not the typical one. And I hope to convince you that it's a new and exciting vaccine. Listeria monocytogenes is a human pathogen, a bacterium that is gram-positive and has a flagella that it allows the bacteria motility. It's an intracellular facultative pathogen, and it behaves almost as a straight intracellular pathogen. There's 13 serotypes, but most of them are coming from isolates of food-related outbreaks. And the two most common and well-described serotypes are the two ones over there, and most of them are coming from the 4B serotype. It is localized all over, in water, in silage, in animal deaths, in food, and it was originally, before being a pathogen, a saprophyte. What about human listeriosis? Human listeriosis is a low-incident infection, at least it was till 10 years ago. There's 1 or 3 cases per hospital per year, more or less per 100,000 inhabitants, so it's not too many people infected with listeria. But the problem of listeria is that those people that get infected, it has a really high morbidity, almost 70 to 80%. And whose are those? Well, the risk groups are mainly the elderly, neonates, pregnant women, immunocompromised patients such as cancer, liver transplanted patients, and autoimmune disease patients. Why? Well, you get infected with listeria because the food is contaminated, mainly cheeses or meal-related products, and disseminates quite good in the bladder, and from there it expands to the lymph nodes, and then from there to the blood. And in the blood, it will go to 2 organs, either the liver or the spleen. And in those 2 organs, you clear up the infection. If occurs that the infection is not clear up in the spleen or in the liver, then you will have a big problem, because it will expand through the blood to the brain or the placenta and will infect the fetus and will cause abortions. So which are the terrible diseases caused by listeria? So mainly meningitis, septicemia, encephalitis, abortions, and preterm deliveries. The transmission, as I said, it was with the food. Is quite rare by contact, but some veterinary people and people working with animals, especially cows and sheep, they get also contaminated with small injuries in the skin, and also, of course, the mother-fetus transmission. It has a high tropism for the placenta and the fetus, and for the central nervous system. And the treatment is, of course, a bacteria with antibiotics, but the problem is that it's difficult to assess where listeria is, that it will be in the brain or in the fetus. It is difficult to calculate the dose of those antibiotics, and it is difficult to calculate the schedule, especially for the unbirthed fetus, because you have a short time. And the cells that are the main targets of listeria are monocytes, macrophages, epithelial cells, endothelial, or dendritic cells. What is the cycle of listeria inside these phagocytes? There's a number of virulence factors that are good candidates for vaccines for listeria, and all of them has to do with the typical intracellular life of listeria. There's 2 internalins that are involved in the invasion of the pathogen. Then once it's inside the cell, it goes to a phagosome. In this phagosome, Is this thing working? No, jeez. Okay. When it's in the phagosome, it will stop the maturation of the phagosome, and there will be virulence factors that it was described by our group, that is the glyceraldehyde 3-phosphate dehydrogenase, GAPDH. And after that, there's a pore-forming protein that is the main virulence factor and the immunodominant antigen of listeriosis, that is listeriolysin O, that will make pores in the membrane of the phagosomes, and then the phospholipases will help to dissolve the membrane and allows the pathogen to go outside to the cytosol. And in the cytosol, there's a protein that nucleases actin filaments that is called AT-A, that is quite important because it forms a comet pole that allows the bacteria to inject into the other cell without touching the extracellular environment. So it will infect cell to cell and spread cell to cell without even being seen by the immune system. So from all of these virulence factors, there are mutants of bacteria deficient genetically in these genes, and most of them will be attenuated, like for instance the listeriolysin O or the AT-A. But from the GAPDH, the mutants will be lethal. So what about the immune system? Immune system against Listeria is first a really strong innate immune response involving mainly phagocytes like macrophages, dendritic cells, or PMNs, and also NKs. And afterwards, there will be an adaptive immunity. Well, that will be quite important to really clear up the whole infection and to develop immunity. Important, of course, to develop a vaccine. And it will be involve B cells and T cells. But even when B cells are important and antibodies are produced when you are infected with Listeria, they won't be important at all for any vaccine because those antibodies are not giving any protection, and it is totally a T cell mediated immune response. So everything you have to do is to prepare a vaccine that is totally inducing T cells, both CD4 and CD8. But it is much more important the CD8 than the CD4. And I will show you the data that we have with that. Well, what is about the status of the vaccine and the epidemiology today, at least in our country? This is data from our country in Spain. Well, the cases of listeriosis has increased tenfold in the last 10 years. And that means that from being just a really rare pathogen, right now is a medium incidence pathogen. We have right now 14, 15 cases per year per hospital in Spain, that it will mean per 100,000 inhabitants. So when we have analyzed these new cases that really surprised us, in fact, last year we have in the north of Spain, where I live, an outbreak with 18 patients. What we have observed is that it's not a problem with the pregnant women or with the neonates. They have the same incidence as they have before 20 years ago, but the problem comes with a higher incidence in the elderly and especially immunocompromised patients, especially cancer patients, and especially liver transplants and several autoimmune diseases. Which are the causes? Well, we came up with 4 or 5 different causes. The first one is, of course, a change in diet. We have changed our diet, especially in North Europe, from a really prepared everyday meal to an already prepared meal with long storage. And Listeria is quite clever in this long storage food because it can grow quite nicely at -20. So if you have just one bag in one food, with 6 months, you will easily have 1,000. And 1,000 will be a really big problem for a pregnant woman. So there's also a clear chain in the bacteria and immunity. You have to think that Listeria is also a microbiota, so we have a natural immunity against this bacteria, or at least we have it before. But right now, the new strains that we have been analyzing, they are 50-fold increase in virulence compared with the isolates that we have 20 years ago. So that means that when you are healthy, that's no problem. But when you are not healthy, you are a cancer patient or autoimmune patient or a transplant patient, that is a big problem. And the other cause, of course, is the new biological treatment of some of these patients. Of course, the anti-TNF in the autoimmune patients are increasing the patients that get infected with listeriosis. And there's also the good technology for new transplantations. We have an increase in the liver transplantation, at least in our country, for 100-fold. That's also an important problem with listeriosis, because they are getting also listeriosis in the hospitals. And the worst thing is that we don't have a vaccine available for listeria. And the worst thing is that the attenuated bacteria are not giving any protection, and that you require listeriolysin for getting immune response and a good protection against listeria. And there's a new therapy right now using listeria, attenuated bacteria, for cancer patients. And these cancer patients will be, in a while, really requiring anti-listeria immunity. So really, there's a requirement in a vaccine. In fact, Aduro is having almost getting ready a vaccine using listeria, attenuated listeria, against pancreatic cancer, against ovarian cancer and prostate, as well as Advix, with a construct of listeria that it has already in phase 2, and I think these are data from 2012, so right now it's in phase 3 already, the one that they have for cervical cancer and the one that they have for prostate. So these patients will require a vaccine. What do you require to develop a vaccine against listeriosis? Well, there's, like as I mentioned before, there's like two groups, the high-risk group and the low-risk group. The low-risk group will be a general vaccine for all the population, for the healthy people, in all ages, neonates, women, all over, elderly. But the high-risk, it will be like two types. The ones that they are healthy, pregnant women, have a high risk of listeriosis, but they are healthy. And the elderly. But there's another high risk that are the patients, the immunocompromised patients, autoimmune disease, the cancer patients. And you have to prepare a vaccine that can protect to all of them. So which vectors? As I said before, unfortunately, listeria is a T-cell mediated immunity, so you require vectors that will increase the T-cell response. So we thought that cellular vectors will be working much better, especially dendritic cells or macrophages, or vectors targeted to dendritic cells. And also you really have to select your antigens, which epitopes of these antigens, and we have thought in two of them, the listeriolysin and the GAPDH. And that's what I'm going to show you. How do we do all the experiments with this vaccine? For the risk groups, we have chosen two animal models. These are typical mice, the black mice, the B6 mice, that is really resistant to listeriosis. And we have chosen as a model for the low risk group. So to develop in them all the results to prepare for the healthy population. And the Balsi mice, the white mice, are high sensitive to listeriosis, and we have used them as a model for the high-risk group. So to develop the vaccine for the immunocompromised patients. The vectors that we use, we came up with the idea that they have to be safe, and they, of course, have to induce all of them cellular immune response. They have to be non-toxic, without any live pathogen, an efficient antigen-specific response, and a powerful innate immune response. And as I said before, what we have thought is in dendritic cells or macrophages, or vectors targeted to dendritic cells, such as nanoparticles. So the first vaccine, we prepared it in dendritic cells. So the first thing that we checked, whether Listeria was able to infect dendritic cells, activate the dendritic cells, and whether attenuated bacteria that doesn't have the listeriolysing gene was not able to get infection in these cells. As I show here, well, listeriolysing O was responsible for the survival of Listeria in dendritic cells, and also for inducing maturation markers on dendritic cells. So the vaccine vector is a really mature and differentiated dendritic cell. Regarding the antigens, as I mentioned before, that they have to induce CD4 and CD8 immunity. So if you have to do that, you have to think that maybe the best antigens has to be those that will be in the antigen processing compartment. And from those, we choose listeriolysing O and GAPDH because they were the only ones that they were able to induce CD4 and CD8, both of them. So now we tested and looked for epitopes. Which epitopes from LLO or from GAPDH? So we checked that they were in the processing compartments, that they were binding to class 1 and class 2 MHC molecules, and that they were inducing a good CD4 and CD8 T cell response. And here comes the design. We get bone marrow from the femurs of the mice. We differentiated with GM-CSF to dendritic cells, or with just M-CSF to macrophages if we wanted to check the macrophages. And then we loaded with our peptides. We give a dose. This will be our vaccine. We give a single dose with 7 days, and then we came up with our pathogen for just 3-5 days. Listeria is a very fast pathogen that in the 3-5 days you will have all the immune response. And these are the results. Both a really short peptide from the listeriolysing O that is CDH restricted, give a good result of protection. Also the GAPDH peptide that is 22 amino acids on the N terminus also gave a really good result. So the immune response was mainly IL-12 and interferon gamma, and the specific response was both CD4 and CD8. The problem with one of the vaccines, with the one with the LLO peptide, it was that the only induced CDH response and not CD4. And at least in listeriosis, although it is important the CD4 response, you will require mainly CD8. So still we thought that it was okay. The macrophage vaccines we tested with these peptides, and the problem was with them that they got good protection, but they were really high toxic, and we have half the animals dead. So we don't use the macrophages anymore. Now we move with this vaccine that was working with the dendritic cells, we move to the nanoparticles. Why nanoparticles? Because these nanoparticles were tested to be targeted to the dendritic cells. And they are gold nanoparticles. The core is gold, and they have different carbohydrates. And you put the peptide You synthesize the nanoparticles with the peptide, with LLO, for instance, the 9 amino acid peptide from LLO, and you put also glucose in a ratio that you have 9 glucose versus 1 peptide. In this way, you have a nanoparticle that it's really filled up with the peptide and with the glucose. Why glucose? Because glucose is really targeting the nanoparticle to the dendritic cells, and it's also enhancing a little bit the immune response. And what we observe is when you give it this nanoparticle, that is called GMP because it's a glyco nanoparticle, glucose nanoparticle, and LLO and the peptide, what you observe is, well, this is the normal listeriosis in the non-vaccinated animals. You have a huge splenomegalia and a granulomatosis in the liver. And with the vaccinated mice, you don't have the spleens are normal, the liver too. And we also took the small intestine, because Listeria usually mice goes to the small intestine. It's a little different than in humans. But we also tested in order that we didn't have any bacteria in the small intestine. And so far, even given the nanoparticle with the dendritic cell or alone, it was working really good in protection. But the trick was an adjuvant. As I said before, Nick, the trick for the nanoparticle vaccine, it was to use advax. Using advax, what we get was full protection, almost as good as the ones with the dendritic cells, in the low-risk mice, in the B6. We didn't get really high protection in the other mice, in the high-risk model, the BALSI, but at least it's working really good, giving good CD4 and CD8 immune response. And of course, protection against listeriosis. What are the conclusions? Well, these vaccines are non-toxic. The disadvantage of the nanoparticle vaccines are that they are only working in the low-risk group. They are not working in the high-risk group. So what about the high-risk group? So we change to the other antigen, whether maybe we were lucky with the other antigen instead of the LLO, the GAPDH. So with the GAPDH, we perform first a bioinformatics study to check for the epitopes, and we compare it with all the epitopes already known for LLO, both in the high risk, in the B6 mice or in the BALSI mice. So all of those that are known to bind to class 1 or class 2 molecules in those different mice. So the best peptides were two, the 22 first amino acids and the 15 first amino acids. Why? Because they have 4 epitopes for class 1 molecules and at least 1 epitope for class 2. Compared with the peptides that we were using with listerolysine that were either class 1 or class 2. We didn't have those peptides that have both epitopes. So apparently from this bioinformatic prediction of the binding, the GAPDH 115 and 122 will be the best ones. But we developed a method in order to check whether we were going to have success. The first thing was using these different peptides with advax to activate the dendritic cells. And what we observed was that the best ones were the GAPDH 115 and 122, and then those with LLO that were restricted for MHC1, meaning class 1 histocompatibility. And once they are activated, so we thought that, that with these dendritic cells, we have to check the T cell response. In order to have it really fast, what we did was to use the dendritic cells with advax to inoculate the foot pads of the mice. And then you check the swelling. as Nick reported before. And what we observed is that the swelling was really good with the dendritic cells loaded with the GAPDH 115 and 122. And we also get good records with the LLO restricted for class 1 molecules. We thought that this would be a good model to try in humans because DTH response is easy to check in the cutaneous, in the skin. So that would be a way that if we prepare a vaccine for humans, check whether these persons were going to get an immune response, a T-cell mediated immune response or not. When we went to the animals, what we observed is that the best epitopes were the GAPDH 115 and 122. Both of them were really good. We didn't get better results with the 122 that is longer than the 115. And they were better than the LLO, the one that is able to bind to both molecules of class 1 from the high-risk group and from the low-risk group, and better than the one that is only binding to the low-risk group. And the rule, at least for listeriosis, for preparing vaccines, was that at least you have to have 4 intermediate binders to MHC class 1 and 1 weak binding to the MHC class 2. So we think that this new design, it will be good for the cancer and autoimmune patients, and of course for the pregnant women, because it will be safe to have a little bit of blood of a woman that it will be risky for listeriosis and to inoculate with their own dendritic cells. And the good thing of this is that it will go to the fetus. So you could prevent the fetus from getting listeriosis. And this is the T-cell response. I put first the good data that of course, they were protected, and there was the immune response. And that's the immune response. We even get B-cell response that is not quite normal for listeriosis. And the final conclusions are that, well, we have at least 2 different designs. One that we think that it will be working for all the population, that it will be the one with the nanoparticle, and this one that it will be good for the patients at high risk of listeriosis. And what are we doing now? Well, we are preparing right now this nano vaccine with the new antigen, with the GAPDH 115, and we hope that we will cover all the fellows in this way. And these are the people in the lab participating. And thanks to all of you. Any questions?